in the DNA fingerprinting. What if we wrote DNA fingerprinting? Why I need to do the DNA fingerprinting? Like this, whenever you are examining, to understand the child dispute case, rape case, murder case, and to understand the human diversity or any other species diversity, DNA fingerprinting is used more frequently. So Professor Alex Jaffe, Stanford University, started this DNA fingerprinting and achieved much more. Professor Alex Jaffe of Stanford University, right? If you're talking about this DNA fingerprinting, in this we are understanding one particular character controlling allele is having what particular nucleotide sequence that we are understanding. And the differences among all the alleles nucleotide sequence also we are understanding. And then we are solving the issues whatever, right? If you're talking about the DNA sequencing challenges, okay, we are comparing the sets of 3 into 10 to the power of bases, for example, would possess significant challenges. So sequencing this much lengthy DNA is a very difficult task. So that is the reason why we are doing this research work in multiple desk or in the case of multiple level. Okay, from this DNA segment to this DNA segment, we do the research work. Like this, the scientists used to segregate the work among each other, and then they work, and then they will interrelate all the work together, right? The role of the reproductive sequence, okay, is very, very important to understand. For example, uh, X person is having 7 reproductive sequence for the no shift. The Y person is having 8 reproductive sequence for the no shift. The Z person is having 12 reproductive sequence for the no shift. So for the nose controlling DNA length is same for all they would be. But the number of reproductive sequence differs from one person to other person. That we need to understand in detail. Right? Now we are coming to next explanation. Non-coding nature. Repeated DNA sequences do not code for protein. Of course, I already told you not to be repetitive sequence. I am worthless. I can't do anything. Like this, repetitive DNA sequences are performing. It is a high degree of polymorphism. It can't do anything. Okay, it is showing maximum differences with each other. Okay, it won't express this on its own. Right? Now we are coming to forensics application. DNA fingerprinting is a crucial tool in forensic science. We all know very well, right? Uh, Here, by understanding the let's be here, okay, by understanding the saliva cells DNA, by understanding the sperm cells DNA, we can find out the difference. I already told you rape case, murder case, child dispute case. All these cases we can understand by doing DNA fingerprinting at the proper time, right? Paternity that we already told, right? And now we are okay in the same data fingerprinting we can exploit this research work for understanding the variants. You know very well the coronavirus is having many variants. Okay, even now also we're all suffering with many variants of coronavirus. So which variant is very harmful? Further, the specific vaccine is what? So like this we are understanding the specific variants understanding. uh, we are uh, doing it with the help of the technique called DNA fingerprinting, right? So uh, DNA fingerprinting helps us to understand the amount of mutation. DNA fingerprinting will help us to understand how one variant develops from the other variant that we can understand, right? DNA polymorphism refers to the allelic sequence, variation at the locus in the human population with a frequency greater than 0.01%. So DNA polymorphism refers to the allelic sequence variation. What do you mean about that? Uh, Here I already told you the skull cell controlling DNA length is same for all human beings, whereas the number of repeated sequence and the number of nucleotide location differs. How come you are saying like this? There is a reason why one human being is having a big size skull, other human being is having small size skull, other human being is having medium size skull, right? In the genetic mapping and evolution, we are understanding, okay, if this much alleles are existing for a particular character, the probability of getting the character is this. The probability of getting the character is that. Like this, we can understand, right? Now we are coming for DNA fingerprinting technique. Alex Jaffe, I already told you, okay, variable number of random repeats, also called as satellite DNA. There is nothing but a probe DNA. There is nothing but a probe DNA. Either you say probe DNA, either you say satellite DNA, or very number of pattern repeats. That VNTR or satellite DNA or probe DNA will help us to understand who is the culprit. Nearby to the murder area, even if you're getting the particular accused skin cell DNA or the sperm cells DNA nearby to the murder area, any one particular DNA if you're getting, we used to call as alien DNA. It's not exactly the alien. So it not belongs to the murdered body. It not belongs to the raped body. It not belongs to the child rescued body. So some other cells which is existing than the body. That is called what? Probe DNA. That probe DNA will help us to understand who is the concerned culprit who did this mistake. Okay, that is the main advantage of the DNA fingerprinting. So the wide application of the DNA fingerprinting is not only limited to forensic science, we are improvising the agriculture growing variants. We are improvising the microdosm variants, right? That is the way we are working on the DNA fingerprinting. We know very well DNA fingerprinting. One of the important techniques of PCR, polymerization chain reaction technique. We are duplicating the given DNA into multiple copies, right? By doing DNA fingerprinting. Now, before going to the order of replication, in the DNA fingerprinting, order of replication, we are going to study the evolution chapter. Okay, in the next forthcoming classes. Before that, we'll discuss the proteins. Can you see the beautiful DNA fingerprinting here? In this DNA fingerprinting technique, what you need to understand basically is uh, how the bands are segregated. How the bands are segregated. Can you see this picture here? How the bands are segregated. So 1, 2, 3, X, everything is here, right? So which one is that answer? We are taking the DNA. So near the X, uh, here the option shows the possible parent of child X. 1, 2, 3. Okay, which one is the parent? It is we need to find out, right? Okay, 1, 2, 3. Which one is the parent? Which one is the parent? 1 and 3 is the parent, right? How come you are saying 1 and 3 is the parent? Why not 2 and 1? Why not 2 and 3? So why? Because 1 and 3 is the parent. Okay, if you are taking the band similarities, okay, can you see this phrase here? Uh, 1 and 3. 1 and 3 is not having the DNA in the same lane. It's not having the DNA in the same lane. Whereas the 2 and 1 is having the DNA in the same lane. Can you see this? 1 and 2 is having the DNA in the same lane in one place. Okay. And then 2 and 3 is having the DNA in certain places. In certain places. If that is the case, then only 1 and 3 is the correct answer. Right? You should know why 1 and 3 I chosen. Right? Now we are coming to number 2. Again, back the board paper we are discussing. It is one of the fantastic assertion reasoning question. Synthetic oligonucleotide. Oligonucleotide means it is made up of 6 to 9 nucleotide DNA. The primer binds to the uncertain DNA at the complementary region. Okay. So what we need to say is here the assertion is correct, of course. Okay, here the reason is accurate. Okay, reason is accurate. So obviously the option A is the correct answer. Right? Option A is the correct answer. So here, of course, the primer is required for making the new DNA that we are seeing as synthetic oligonucleotide polymer. The primer binds to the uncertain DNA at their complementary region. Yes, of course. Uh, so that is also the correct reason. That is why we choose option A. Now we are coming to question number 3. Write the scientific name of the source of the thermostable DNA polymerase used in PCR. Thermophilus aquaticus. Thermophilus aquaticus is bacteria that is having a DNA polymerase that we are using in this experiment setup. Okay, can you verify whether I told correctly or not? Am I correct? Thermophilus aquaticus, tag polymerase. Okay. So this DNA polymerase of this particular bacteria is doing the is used for PCR technique, right? Now we are coming to question number 4. State the advantage of using thermostable DNA polymerase. But because in PCR we are maintaining 90 degrees Celsius and above, sometimes this get inactivated. That is why you are using thermostable DNA polymerase. That means thermophilic bacteria DNA polymerase, right? We are using it. Related videos, whenever you have time, come to the portal and then use it and study. And now we are coming for examples. In the examples, the sample of DNA is processed with PCR amplifications and gel electrophoresis. Gel electrophoresis. Are you getting my point? So here, PCR amplification is DNA multiplication, right? Explain what is band represents and why they are important in DNA fingerprinting. DNA fingerprinting, right? Uh, so this DNA fingerprinting will help us to understand the issues. That is one thing. By comparing the DNA fragments, how we got the DNA fragment? You added what are the victim DNA you're getting, what are the probe DNA you're getting that you're multiplying into the number. And then what you're doing, you're adding restriction enzyme to it. And then the restriction fragment, whatever you're getting, that you are running the gene medium. Right? There is a restriction you need to understand. In the parent testing cases, the DNA fingerprinting is conducted for a child and potential father. And okay, yeah, the band father at work, okay, this is where base pairs are there. Okay, and then comparing the child DNA profile. So, and then we are saying matches exactly potential father, right? So these are the things we discuss in the case of DNA fingerprinting, right? Uh, now tips for exam. Okay, as I already told you, why the PCR is separately we are discussing, and also we are discussing as a part of DNA fingerprinting. In DNA fingerprinting works on the principle of gel electrophoresis. What do you mean by gel electrophoresis? We are separating the DNA molecule into the charges, right? This is the message you need to understand in the PCR.